Hello, everybody. I wanted to thank you for attending this webinar. Today we're going to focus on an interesting webcast presented by Hagar. The title of this webinar is Rare Case of Early Onset Parkinson's Disease Solved by Whole Genome Sequencing Analysis, which we'll hear her talk about how exon inversion was invisible to exome sequencing. But just a general introduction, here we are joined by a couple of panelists. So we have the VP of Sales of North America, Kim Wohler. My name is Eli Sward, and I am the Field Application Scientist for the genetics application. And then we also are joined by Hagar, who has essentially expertise in the bioinformatic workspace. She works at the genetic department for the Hadassah Hebrew University. And today we're going to focus on a very interesting topic which she has identified within her lab. So first of all, I wanted to thank everybody for joining. If you have any questions during the webinar, please enter them into the Q&A or the chat panel. We'll address them at the end of the webinar. And if we don't have time to address them, we'll also follow up with kind of a direct email. We'll also send a recording of this to all the people that have registered to this webinar. But with that said, again, I would like to introduce Hagar. Thank you for joining, and we're looking forward to your presentation.
Hi, everyone, and thank you, Eli, for the introduction. Today, I'm going to tell you about a rare case of early onset Parkinson disease that was solved by whole genome sequencing analysis. But first, I wish to declare a conflict of interest. I'm currently the product manager of genetics analysis, but the research project that I will tell you about today was conducted and published before that. So just to start with a short introduction on Parkinson disease. So Parkinson disease, or abbreviated as PD, is a progressive disorder that is caused by degeneration of nerve cells in the part of the brain which controls movement. The nerve cells die or become impaired, losing the ability to produce dopamine. Eventually, this causes unintended or uncontrollable movements, such as shaking and stiffness and difficulties with balance and coordinations. And symptoms begin gradually, but worsen over time. As a geneticist, I'm interested in the genetic variation or the genetic contribution to Parkinson disease. So the genetic variation is estimated to contribute approximately 25% to the overall risk of developing PD. The genetic variants related to PD vary in terms of frequency and risk. On the one hand, there are a number of rare variants in single genes that are sufficient to cause disease. Example to this is just genes such as SNCA, PARK7, PINK1, PARK2, which is also known as Parkin. And variants in these genes are monogenic, causing Parkinson disease, and are pathogenic variants. On the other hand, genome-wide association studies have identified a large number of common genetic variants that individually contribute a small amount to the risk of developing PD. In the middle of the spectrum lie variants that are uncommon but are rare, such as in the gene GBA and LRRK2, and these ones exert an intermediate risk to Parkinson disease. The story that I'm about to tell you today is on a consanguineous family of a Yemenite Jewish origin. As you can see here in the pedigree, 4 out of the 12 siblings developed young onset dystonia with a median age of 11 years. So this is really young. The first reported symptom was dystonic foot inversion during walking in 3 individuals, and upper limb action dystonia with tremor in the 4th individual. The dystonia gradually generalized to the other limbs with asymmetrical severity in all the affected individuals. Well, all of them were dramatically improved shortly after oral levodopa. So we classified it as a levodopa responsive PD. We performed exome sequencing first to one affected sibling, the oldest, but it was without a diagnosis. So we actually performed 8 exome sequences in total, 4 affected individuals, siblings, and 4 healthy siblings. We only found one shared rare homozygous variant between the affected individuals, and it was in the gene named TCP1. It was a missense variant. Well, we further segregated this variant in the rest of the siblings that were unsequenced by the exome, and TCP1 did not completely segregate it in the family, as one sibling that was considered to be healthy, V5, was also homozygous for this variant. Although we found that it wasn't segregated completely in the family, we decided to use gene matcher, which is our common method when we just want to understand if there's another family that could may be relevant to this story. So we put the TCP1 in gene matcher, and that was actually very encouraging. We found a hit with another consanguineous Persian family with 3 affected siblings, and they all presented gait disturbance. And this was actually a different missense variant in the same gene, but it was approximately to our variant. So then we thought, okay, let's check again V5, the sibling that was considered to be healthy. And the clinical evaluation of this individual revealed right wrist lead pipe rigidity and neurological examination. So we thought maybe the sibling was too young to show the presentation. Maybe it was a more mildly affected with a disease. And this is why we initiated a collaboration for functional studies, and we generated C. elegans using a collaboration with Dr. Ronen Zaid-El-Bar from the Tel Aviv University. And what he performed was C. elegans for both variants, our and the Persian family variant, using CRISPR-Cas9. So this was the best functional studies that could have been performed, because we actually integrated the human missense variants into the worms. So this was the control C. elegans, and we were actually hoping to have in the affected muted worms some shaking worms, but actually they were completely fine. So we couldn't find any difference between the control group and the mutated worms. So then we went back to the family and we performed linkage analysis using the exome data that we already had to the 8 siblings, again, 4 affected and 4 unaffected. And what we did here, we searched for a homozygous block in the affected siblings. And looking deeply into chromosome 6 block, where the TCP variant is found, we actually noticed that the Parkin gene is found exactly on the same block. And this was rather encouraging, because we know that biallelic pathogenic variants in Parkin lead to early onset Parkinson disease. At this point, after we rule out any copy number variations in Parkin and single nucleotide variation, we immediately knew we need to do whole genome sequencing. And that was very nice, because whole genome sequencing using the Genix analysis platform has identified an inversion of exon number 5 of the Parkin gene, followed by a common 77 kb deletion. So what happened here was actually an inversion with a deletion inside. And this was in the platform, was the first probe, because that was the best candidate considering the high correlation between the gene, the Parkin, and the clinical presentation of our patients. And we, of course, we validate that. So what happened here was just an inversion of exon number 5, which is totally balanced. So we're not expecting any copy number variation in these patients. And we actually validate that also on the cDNA level. And we found that there was an intronic inclusion. So some arbitrary sequence from intron number 5 was inserted instead of the exon number 5. And when looking on the conservation of the deleted exon number 5, we found it to be highly conserved, explaining the pathogenicity of this variant, which probably causes just a complete loss of function. This work was conducted in a collaboration with Professor Tamar Harel and Dr. David Kadir. And up until now, no copy neutral inversion in the Parkin gene was published as disease causing. And when we went back to the data, we found out that V number 5, the individual that was considered to be healthy to begin with, had a parental recombination, excluding the pathogenic Parkin inversion as homozygous in this patient. So while he was homozygous for the TCP1, he was only heterozygous for the Parkin inversion, which really makes sense why he's healthy. So overall, we spent 2 years of work. We did 8 exons, 1 animal model for 2 missense variants for the wrong gene, of course. And we performed only one genome sequencing. And we saved that. All that work could have been saved with just to start with a whole genome sequencing and analysis in Genix using the Genix platform had revealed in just a couple of minutes the diagnosis for this family, which of course enabled later to perform PGD in this family. So this work was published in Brain, and of course you can further, those who wish to have more information can further found the paper and read it. And again, I just want to say that this was a collaboration with Professor Tamar Harel and Dr. David Kadir from the genetic department at Hadassah and also at the neurological department. We really think the take-home message here is that we think that whole genome sequencing will solve many cases of early onset Parkinson's disease. And I highly recommend, before conducting any functional studies, just to perform whole genome sequencing with a high quality analysis, just to make sure you don't have just a straightforward diagnosis in your patients that is invisible to the exome analysis. And with that, I would like to finish, but just give a thank you for the genetic department at Hadassah and my bioinformatic teams, and to Genix Analysis for the platform they have created that enable to solve this case. Thank you.
Great. So I just wanted to say to Agar, thank you for providing that presentation. We're definitely, if anybody from that attended is interested in the links from her publication, we'd be happy to send those out, after the particular link. That said, I know she kind of, Hagar went into some detailed information. If you have any questions about what was presented there, please enter them into the chat panel or the Q&A, and we'll address them at the end of this. Meanwhile, what I wanted to do is just kind of give a brief introduction into the application that she was using from a field application scientist perspective, because I know a lot of the attendees probably haven't seen the GenX application, or maybe want to hear about some of the newer features. And so what I'm going to do is just go into a couple of slides on the application, and then after I finish, we'll go into some of the questions. So with that said, let me I'll just jump right into some of our applications here. So the GenX Analysis, really, our company, we provide 3 different kind of modules. First, what Hagar implemented was the GenX Analysis, which is used for NGS analysis and interpretation. But ultimately, we do have other applications, which include the GenX clinical applications. So these are kind of off-the-shelf, ready-made reports, including pharmacogenomics, carrier screening. Basically, if you want to implement a protocol and generate a clinical report with your existing NGS data, these are available to our users. And then we also have GenX Discovery, which is utilized for more of a research-based approach. So if you wanted to identify kind of novel findings for associations related to particular phenotypes, this is going to be a great resource, which we're currently developing. But ultimately, we're really trying to automate this diagnostic processing from sequencer to report. We incorporate machine learning and AI-based approaches, but we also want to create a resource for discovery. And so to talk a little bit about what the GenX software offers, really it is an end-to-end solution. So we can start from the FASTQ or raw data all the way to a clinical report. But there's a lot of steps in between that are very customizable and integrated with your current bioinformatic pipeline. So to talk a little bit about the raw data processing, so we have simple integration with any particular pipeline that you're wanting to run. And you can start with either the FASTQ or VCF files. For those that maybe are interested in large sample workflows, you can also perform a batch upload. Basically, you can run it from a TSV file. Once you start processing, you can close down the application. When you come back, you'll get a notification that that sample is processed, and then you can begin your analysis. But we do support fast annotations as well as the processing. So for gene panels and exomes, you can expect to basically perform an analysis from FASTQ in under 30 minutes. And then for genome, that's under 3 hours. So this allows kind of a rapid processing from FASTQ to a clinical report. In terms of the flexible workflows, so one of the questions that we get is, is your filter logic locked down or can it be customized? So if you want to obtain repeatable results, you can create a particular filter logic. You can implement it into a protocol and use that for all downstream analyses. We also support ACMG and AMP guidelines. If you create an interpretation for a given variant, it'll be saved and stored. So if you were to see that variant in a future sample, you'll be presented with your interpretation. But we do have really nice audit trails from an account management perspective. It is a great resource. We're also not a black box solution. So if there's any capabilities that you're wanting to see, it's very transparent and it's easy to work with. In terms of integrations, we support both cloud-based as well as on-premise. So, depending on if you have to be behind a firewall, we can support that infrastructure. We also have questions on whether it can integrate with LIMS or electronic health record systems. We have excellent API documentation to support these integrations. We also have reporting capabilities. And then if you're interested in security, we are HIPAA and GDPR certified, and we're backed by our ISO certifications as well. And then reporting is obviously very important. A lot of companies like to outsource these to third-party environments, but with GenX, this is all supported in-house. So if you wanted to customize these according to your lab designs, you have that capability. And then the reports can be automatically generated and pull in a variety of information from our evidence categories. So in terms of our key differentiators, I won't spend too much time on this because, if you wanted to schedule up a follow-up meeting, I'd be happy to kind of deep dive into this. But our phenotype incorporates tree-based search terms as well as HPO ontologies, and it scans biomedical knowledge bases to identify associations between the gene and the phenotypes that you've entered. From a clinical geneticist perspective, and what Hagar likely used for her CNV or her structural variant interpretation was the phenotype algorithm. So you can do a deep dive investigation, but you can also see a prioritized list according to that disease phenotype. We also integrate a non-clinical phenotyper. So if you want to incorporate additional information for novel diseases, we have incorporated this to include expression profiles, animal models, as well as other databases. So this is available to our users. We also support the ACMG classification. We automate 8 or 17 of the 28 ACMG criteria. which allows you to work with the kind of the best practice guidelines. You also can modify these, which will be stored for all downstream interpretations. And we also stay up to date on our annotations. So for a lot of kind of those that are familiar with the in-house bioinformatic pipelines, these annotation sources are updated pretty regularly. We stay on top of this through an automated process, and our annotations are updated on a monthly basis. And then lastly, and maybe most importantly, we have whole genome sequencing analysis. So as Hagar presented in her talk, there's situations where exome sequencing maybe doesn't identify your candidate variant, and so you need to apply the next level. GenX analysis supports one, rapid annotation, but also the ability to deep dive and get a better understanding of maybe a causal mutation. And in this case, it was a structural variant that was associated with an inversion. So GenX analysis supports the ability to analyze copy number variants, structural variants, as well as repeats. And one of the great tools is the ability to compare your CNVs with your SNVs that are coming in from your VCF file. This can be a line of supporting evidence to say, if we're looking at a homozygous deletion, we would expect the variants to have an allele frequency of maybe 0 or 1. And you can either confirm that that event is real or disregard it as a false positive. Furthermore, we also support enhancer analysis. So if your CNV impacts a region outside of a gene, such as a regulatory element, we provide a variety of information in terms of association between enhancers and that gene and phenotype. So this is a great tool, and then you can also visualize this as well as perform the focus SNV analysis within your region to determine, one, if there has been any candidate variants observed, and also if they are observed at a high classification according to the ASMG guidelines. So with that said, just to kind of bring this full circle, obviously our GenX application has been implemented through a variety of different hospitals and research institutions, but in terms of scalability, we like to reference Bambino Gesù, which is the largest pediatric research center in Europe. With the application of GenX, they were able to increase the number of cases that they analyzed from around 100 per month to around 3,000. So with this rapid annotation process that's implemented with GenX, it allows a scalable pipeline. So it reduced the turnaround time, to around 95%, and then it also increased the diagnostic yields to around 65%. So with that said, one of the things that we like to highlight again is where we are and then where we're going. And so at present, the kind of industry is focusing on whole exome sequencing, but obviously we're transitioning into that whole genome sequencing to identify kind of a greater understanding of the biological impacts of genetic and genomic data. One of the things too that we're beginning to see is the transition from short reads, which is around 250 base pairs, to long reads. So looking at kilobases as opposed to base pairs. So this enables improved alignment, improved mapping, and we've already been involved in pilot studies that has shown GenX to outcompete our competitors. Additionally, transitioning the GenX application from individual analysis to maybe cohort to identify those key those key candidate variants that are maybe associated with a large cohort of individuals. Obviously, this has a lot of applications, and so we're looking forward to kind of deploying this. And it's already available, but we're going to continue to improve on this. And then also pediatric rare diseases to a range of medical conditions. And then lastly is transitioning from strictly genomics to also multi-omics. So we're currently in collaboration with a lot of great companies, and you'll likely see this developed within future releases of our application. So to kind of bring this full scope, so what we're doing is we're essentially providing you with an end-to-end solution, and we are also incorporating all of the different bioinformatic pipelines. So whether that's sampling, purification, taking this to sequencing, data integration, and report, really we're providing you with the ability to harness all of the genetic data, integrate this into your LIMS or EHR system, but more importantly, resolve kind of those novel disease variants that you're seeing within your patients. So with that said, I wanted to thank everybody for attending and for all of our current customers, but we do have a couple questions that I wanted to address here. And so maybe we'll get our panelists back on. First and foremost, thanks again, Hagar, for
Thank you.
Yeah, for giving us this nice presentation. We do have a couple questions here, so let me pull it up. So it looks like one of the questions was, what was the outcome of the family from Gene Matcher?
Oh, the other family, I think the Persian family. Yes, so we had a positive hit with another family, as I've described in my lecture before, but the research group decided that the TCP1 is the pathogenic variant in their case, although the worms didn't show any phenotype. And I think they actually conducted additional studies using different animal models. So this actually may hint that the TCP1 can contribute to the overall phenotype. But I do think, and we highly recommended them as well, to perform whole genome sequencing for the same family, because they might also have a structural variant in the Parkin gene, which is invisible to the exome sequencing. And as far as I know, they did not conduct a whole genome sequencing for the family, for the affected siblings.
Okay, thank you for that. The other question that we have was, how do you filter your whole genome data?
Okay, so this is really a challenge, because when you do the analysis of whole genome, it's much harder than when you do whole exome sequencing analysis, because the amount of data is much broader, and many more information and data that you should relate to. It's not only single nucleotide variants or copy number variants, you should also analyze the structural variants and the repeat analysis. So I think using the system, we have a few tricks that we can play. So we have the allele frequency of the in-house database. This eliminates all the background, the noise that we see, the artifacts. So once I see a positive or a false negative event, which according to my analysis is just more than, for example, 10 times, 15 times that I've seen before, then I can just deduce that this is not a real event. So this is how I call the negative false events. We also have in the system the ability to compare copy number variations to single nucleotide variations. This serves for two purposes. One is just to understand if you have a compound heterozygous, but also just to understand if the event is real, because if you have, for example, a heterozygous deletion and you see a heterozygous variance inside the same position, the same genomic position, then you can immediately deduce that this is not a real event. So this is just another way to really try to investigate the data. And of course, I can elaborate more on a personal meeting if anyone wish to have some more information.
Yeah, and that also applies, if you're interested in scheduling a demonstration or you wanted to get more information on how to perform whole genome sequencing analysis, you can reach out to support@genx.com and we can schedule, a one-on-one meeting and take you through that information. So there's another question from one of our candidates here, and it says, how does your software pipeline compare with Illumina Dragon or the Grail system? So the Illumina Dragon is actually a secondary pipeline that has been implemented within the GenX application. So you can, basically pull in all of the quality metrics coming in from that pipeline into the GenX application and then use that for filtering. I'm not familiar with the Grail system. Have you heard of that, Hagar?
I think it's the Grail system is a blood test. So it's a somatic analysis of a blood test for cancer patients. They run their own pipeline, but we're quite open. So if you have any secondary pipeline that you want to implement inside GenX analysis, this is definitely an option. So.
Okay. And then there's also another good question. So it says, has any transcriptomics or proteomics been done with C. elegans in relations to Parkinson disorder? Just wondering what if it is the best model organism one could be using when modeling this disease?
Okay, so this is actually a very good question because we debate on that a lot. And according to the literature, the worm offers several advantages of the study of Parkinson's disease. And besides the ease of the genetic manipulation, here we use CRISPR-Cas9, which is very fast. The experiments are very fast, the costs are low, but besides that, the worms have been shown to exhibit multiple phenotypic deficient, including, for example, loss of dopamine neurons, disruption of the dopamine-dependent behaviors, increased sensitivity to stress, age-dependent aggregation. So this is actually a common model for Parkinson's disease, and accordingly, a number of C. elegans models were created, including the transgenic worms that express alpha-synuclein, or LARC2, which is one of the risks variants for Parkinson's disease. And also there is a worm for the parking deletion. So yes, basically, it's actually a good model and very easy to generate.
Great. And I'm sure too, we'd be happy to send you any documentation, relevant links to answer that question further as well. So we do have a couple other questions that are a little more specific to the particular workflow. And one of the questions was, how did you validate your structural variant?
Okay, so I think I've shown only one slide on this. The paper has a lot of additional information if anyone wants to have like an additional information. But basically, after we identified the breakpoints using the Genix platform, we extracted the reads that showed the breakpoints, the inverted sequence. And once we had the sequence of the reads, we could just molecularly validate it using just a standard PCR, the breakpoints. So this is how we validate it on the DNA level. And after that, we also purified RNA from one of the affected individuals, one of the affected siblings, and just performed the same analysis using the same breakpoints just to show that the exon number 5 is not inside the transcript, is not inside the cDNA. So this was validated on both levels, DNA and cDNA.
Great. So it looks like that answered all of our questions. Again, I wanted to thank Hagar for giving an excellent presentation.
Thank
for
you,
those
Eli.
for those that attended, if you have any follow-up questions, feel free to reach out to support@genx.com. Again, we'll send this recording to all the people that registered. And thanks again for attending. I hope everyone has a good day.
Thank you.
All right. Thank you.